All right, so while we're all taking seats, for those of you who have not participated in these workshops by the committee, we try to spend a lot of time on discussing what we've heard. So these workshops are not only about presenting science, but also about answering particular questions. And the questions for this session, you can see on your screen right now, and the panelists can see them on the TV monitor in front of them. So the committee comes up with framing questions to take this into more of the practical application, because the committee is environmental science for emerging science for environmental health decisions. And while we all appreciate the beauty of basic research, a lot of our participants are in applied fields, and many of them work for different regulatory and other related agencies. So we're trying through this discussion to perhaps flesh out the interactions or potential applications of the basic research to the actual use. And here on the panel, we'll have one of our government liaisons, Dr. Carlson Cerniglia. And Carl, maybe you can introduce yourself a little bit before we go into questions.
I'm Carlson Egli. I'm Director of Microbiology at the National Center for Toxicological Research, which is part of the U.S. Food and Drug Administration. And just as a bit of a background, I've been in this field, I guess, similar to what Dr. Wilson mentioned, in the early days of doing a lot of metabolism in the 1980s, enormous amount of studies on the metabolism of intestinal bacteria on a wide variety of compounds, mainly environmental toxicants. And now we have a pretty robust program involved on the toxicology risk assessments in terms of the impact of the intestinal microbiota on a wide variety of chemicals, and also the chemicals on the bugs. So that's just a brief.
So we would take questions from the audience and from the web as well. We'd like to get the discussion going. We'll try to stick to these questions, but we can, of course, expand to other things. So for those of you who are interested in asking questions related to the questions on the screen, please line up to the microphones, and then we'll take questions from the audience and the web. But before we take questions, I wanted to really ask our panelists to reflect on what they've heard this morning, and we've tried to frame this into slightly different groups, because there's a lot to noodle on, obviously. And one of the first questions is, how big is the effect of exposures, whether these are drugs or environmental chemicals? And if we can for a second exclude antibiotics, the most obvious ones that are there to kill, is there a major effect, and how long that effect persists? And I perhaps want to start with Dr. Lu, because some of the data that he showed was clear effect, in weeks 1, 2, and 3, but by week 4, there was absolutely no effect on arsenic metabolism in the, microbiome modulated mice. So, have you seen similar effects, in your other studies, and how can you reflect on that when toxicity that we're worried most is the chronic toxicity, and some of, the microbiome is so plastic? So what type of information we have right now to actually think about the magnitude of response to the human microbiome or animal microbiome community by environmental exposures?
Yes. So we also observe in other chemicals, because the gut microbiome change actually a dynamic process. So particularly if you use the animal model with the bacterial infection, because the bacterial infection, the colonization and the interaction between the gut and microbiome is a dynamic process. Okay, so usually in some species, after a few weeks, that's you're going to see the colonization of the bacterial species you put in the gut is going to reduce. So as consequently, you're going to see the whole gut microbiome community structure is kind of balanced back a little bit. Okay, so which is drive the something we observed in the arsenic study. And but for the in terms of the chronic long-term exposure, we done some 3 months, some 6 months study. So that seems the effect is more dramatic compared to the 4 weeks or 2 weeks study. So the gut microbiome, once it's changed over the longer time, it's harder to move back compared to the short-term exposure.
Do other speakers who have experience with this would like to weigh in on the plasticity, and especially obviously it is plastic, but is it really recolonizing quickly, dealing with the exposure, or the exposure effects persist over the entire duration of treatment?
Okay, if I can be slightly controversial. If you change the microbiome, but it does the same stuff that it was doing before you changed it, does it matter? I mean, all the rats that I showed you had different microbiomes in the different cages, but metabolically they were the same. So part of the risk assessment has to be, okay, I've made a change, does it matter? I think the other thing that needs to be considered very importantly is that we know that the microbiome is much more plastic in neonates and young children, and your ability to restore your microbiome gets much worse as you get older. So people my age are beginning to lose their ability to reprogram their gut microbiota back. So it's that sort of question. I think there has to be a risk, but what's the hazard?
Yes, I agree. And to follow up on that, what we've been finding in these toxicology risk assessments, when you really start taking a look at what's been done, which is outstanding work that we've heard today, and certainly many people in the audience and around the world have contributed to this field. But what we struggle with sometimes is this kind of, and again, it gets to your point, I think, this whole quantitative metrics of a disrupted microbiota, and how does that connect to a clinical impact? I think that's the problem we have, because you generate an enormous amount of information, and that's another challenge we have from a bioinformatics standpoint of how we handle it. But that's it.
So perhaps one other way to rephrase this, and this was one of the questions we got from the web, and perhaps Professor Wilson, you're the best to answer this. Other examples or studies of human microbiome with drugs at therapeutic doses that do not have adverse effects, especially the ones that are taken chronically. Is there truly a modulation of the microbiome by drugs at therapeutic doses that are not cancer drugs, that are not antimicrobials? Does it matter or not?
Okay, well, the honest answer is there may be, but I don't know. What you can do the thought experiment and say, well, all right, let's suppose I'm taking a statin. That may well modulate cholesterol metabolism. That may have impact on bile acid production. That will have an impact on the microbiome. Maybe. I mean, just modulating bile acid metabolism doesn't necessarily affect things. You could do that sort of work in animals. I think even animals, given a human microbiome, but you would then have to think very carefully about some of the cage effect things that I pointed out in order to make sure you didn't get the wrong answer, or rather you got the answer you were expecting and then just sat with it.
And Dr. Redinbo, in your push to move some of your compounds into more of the clinical arena, what can you tell us about the length of the effect that you see on the enzyme kinetics in bacteria? Is this truly an effect on the bugs that are there right now when they have the inhibitor, or is this is more of a long-term change that you have, change in the microbiota and phyla of the bugs that you have by affecting a particular enzyme?
Yes, so we've done not long-term studies. We've done fairly short studies, but what I can share with you that we haven't published is that if you treat animals with the inhibitor, the composition of the GI changes. It changes within 24 hours. That change is maintained over a few days of treatment time. And when you take the inhibitor away, each animal's microbiome changes back to the beginning state. And so I see That's why I described it as a vast and dynamic organ. It is really remarkably dynamic and resilient. Now that's a very limited study. We've done this and I'm really sensitive to what Ian said about different strains of mice in different cages. And so we've done that study in a couple different places, but we need to do that more. I do want to just chime in a bit on Ian's point about, does it really matter what bugs are there? And we think about this a lot. We think about the microbiome as being this library, or maybe in a better way, the playbooks of all the NFL teams. And they all might have slightly different covers and some different variation, but they probably have very similar plays in them. And it doesn't really matter whether or not the Falcons are running a screen pass. I'm sorry, Ian, you don't understand this.
I understand enough.
Yeah, okay. I was on sabbatical in the UK, so I could turn this to football if your football. If you
Cricket would be better. That would
okay.
really be better.
I'm quite hopeless there, but let's say you put in a spin bowler. So it doesn't really matter, who's running the play. And I think to your point, that is so really right on, because it can be a very different set of bacteria. And they're probably going to be doing very similar things in a normal, not dysbiosis or disease state. And so I would argue what we need is a set of standardized compounds we can give to animals or even give to humans, and then be looking for biomarkers and metabolites. And so then we could certainly know what their composition is, but we don't really know what plays they're running until we see the outcome of the plays. Do they have certain metabolites at certain levels? And I don't know that we're there yet, but it feels like we're close. So I sort of wanted to suggest that as maybe an action item.
Yeah, certainly that's a very interesting point. And I also wanted to bring Andrew into the discussion. And especially you mentioned FXR and bile acids. And there's, at least to my knowledge, there's very interesting literature in alcohol-induced liver injury and fibrosis and other phenotypes where the role of microbiome is being expanded beyond just contributing to, LPS going into the liver and, sensitizing hepatocytes, where now there is evidence that a feedback loop in reabsorbing bile acids, whether you have, cholestasis in the liver or not, as a component of the treatment, there's a feedback loop through, FXR and other things where now we're trying to close the loop rather than it's just the gut is there to help us or hurt us with metabolism. Now all of a sudden there are feedback loops into the pathology and the actual mechanisms in the tissue. So do you see that as the kind of, the next frontier in trying to measure the impact of the exposures on the microbiome through true mechanistic research in tissues rather than just simply, toxico or pharmacokinetics of chemicals?
Absolutely. I was talking to Lee Kaplan. He's a fatty liver expert, surgeon, and he put it a very interesting way. He was talking about bile acids and said, in our showers at home, we have, a few soaps. We don't have hundreds of soaps or detergents. So when you look at actually what the bile acid makeup is in our bodies, there's hundreds of them actually. So the question is, to pose to everyone is, are they serving simply as detergents? More than likely, they're likely serving as some kind of small, low abundance hormone that is signaling back and forth between the liver and the gut. Now, the microbiome itself can capitalize on this and allow or modify their environment according to which bile acids are there. So they can turn pathways on or off, and it's been demonstrated by collaborations from my lab with Frank Gonzalez's lab, other labs as well. The real question that remains in terms of fatty liver disease, and I'd say it's a bit controversial, is should we turn these pathways on or off? I think it's something that's not yet quite understood. However, where you turn it off is really key. If you turn it off in the gut, you see beneficial effects. If you activate it in the liver, you see beneficial effects. So this is something I think will only emerge as we start to understand these pathways a lot better.
So we'll take a question.
Risha Putzreth, Navy Marine Corps Public Health Center, and of course my comments are my own, not the Navy's or DOD's. There seems to be a dearth of long-term studies, and I was wondering whether anyone has thought to approach the agencies that run cancer bioassays to see whether it would be possible to get microbiome data throughout those studies where we not only have controls, but more than one dose, and they're dosed for the major part of their lifetime.
Your NCTR. Did that ever come up at NCTR? NCTR is running to your cancer bioassays all the time. Did you guys ever, thought of partnering with them on taking stool samples throughout?
There's always an educational piece involved in this when you're in a toxicology world, and then all of a sudden, we've been preaching for a long time, the importance of now the light switch is on, so it's part of our assessment. So thank you for that question, and certainly in our own group we do have relationships with NCI on doing studies on the microbiome.
So speaking of models, and both animal models and in vitro models, and that's the second question. So we've heard today a lot about mouse, rat, some of the human studies as well. I think Matthew, you've elegantly portrayed some of the in vitro models that may be coming up. So what is your perhaps experience thus far with in vitro and trying to modulate perhaps some of the human microbiome and see what effects it has without actually having to go the clinical trial route? So can you comment on that, and then perhaps others have experience with that as well?
Yeah, so we're working hard on this, and we've had some successes and some surprises. I'll paraphrase a quote I heard at a meeting a few years ago, that all models are flawed, but some models are useful. So that's the approach we take into this, and we have done as much as we can in vitro, but still see surprises there. And we realize that a lot of that is because any reduction from the system eliminates things that are necessary. And yet we still feel like if we do it with our eyes open and really mindfully about what we're setting aside, we can find some pretty profound effects. And I think that's got to be the way to go forward with this. As much as I'd love to charge into a notobiotic stance with everything we want to do, it's not feasible, it's not really appropriate, it's probably not ethical, it's almost certainly not ethical. And so I think you have to work it up in a way where you have the in vitro and the ex vivo really established well, because then once you do the in vivo, you have to go back and you have to reset up the in vitro and ex vivo situation, knowing what you then know, and go through that cycle a couple of times. And I'm quite enthusiastic about the utilization of the animals more sparingly, and we're working very hard on that to try to set up systems where we can recapitulate some of the dynamic across the domains of life appropriately, with the limitations of certain models, but more appropriately than maybe has been done to this point. And I think that is probably not, exciting, but I think its impact will be exciting, and so we hope to try to bring that forward.
So, and Dr. Lu, perhaps, again, you mentioned some of your studies with arsenic metabolism and, other more long-term studies that you're doing. So have you started looking beyond just the metabolism and the kinetics effect of the microbiome on the chemical in question? Have you started looking at some of the, toxicity phenotypes? Again, just going back to what Dr. Wilson was saying, does it matter? I think that's the big, question that the regulators and the practitioners really have. We can study all of it, and it's fascinating, and it's probably much more diverse than the human genome, but does it really matter?
Yeah, that's, I actually agree with Dr. Wilson. So, the gut microbiome change is always a change. So the fundamental thing you have to address is whether there are any functional change associated with the community structure, because different bacteria, they can do the same thing. Okay, so and also we need to move forward from the bug to the host. For the arsenic study, we actually also start to look for the toxicity phenotype. We also we start to look for whether there's link with the intestinal permeability, and also the metabolic difference, I mean, in the host, not, I mean, beyond the microbiome itself.
With arsenic and in animal models, I mean, as you obviously know, mouse is a bit of an outlier where you can't really get cancer with arsenic in a mouse, and unless you do the prenatal and maternal exposures. A beautiful work out of NIH's Dr. Walke's lab. So, in your particular experience with the arsenic, what type of phenotypes you're trying to look at, and maybe other chemicals? How close those are to phenotypes or pathologies that are actually going into regulatory decision making?
We actually look for the chronic inflammation as well as some cardiovascular disease endpoint. We haven't, started look for the cancer endpoint on those animal model.
And Andrew, do you have anything to add on to the actual pathology and kind of modulating toxicity by modulating the microbiome? Are you doing any of those studies, or are you aware of those studies that again go beyond pharmacokinetics of the chemical?
Sure. I think one thing we've always been interested in, that is try to understand kind of what the longitudinal profile of a person looks like by a metabolomic approach. I'm not specifically implicating the microbiome here. If we were to take snapshots of everyone, it would be very difficult to discern who's who. However, over time, we could basically build patterns that we would be able to identify you based on a simple metabolomic profile. What's part of that are things like diet, of course, but also your microbiome is going to contribute to that quite substantially. And this is from simple things like urine or blood that can allow you to develop this fingerprint. So the key is, I think it's kind of getting to your question, is that it's not just simple snapshot type experiments where we need to look at a single time point. Really what needs to be done is more longitudinal type approaches where you can see how these interactions change over time. And I kind of got turned on to this book, in conversations with Peter Turnbull, and this is Biochemical Individuality. It's a book written by Roger Williams several decades ago. And essentially, this is this idea long ago talking about precision medicine or personalized medicine, that we are ourselves and not like anybody else, and the things that dictate this are a lot what we've talked about today.
Yeah, if we can follow up at least a little bit on that, because I think the important distinction here is between personalized medicine versus making decisions about environmental exposures and trying to protect most of the population rather than trying to do personalized risk assessment. So in this regard, if the only answer is to take a lot of snapshots, do you think that that's really a feasible strategy forward in a kind of population sense? Because our animal studies and a lot of our human epidemiology studies are taking snapshots, and we've learned over the last 50 or 100 years how to make decisions that are not completely wrong. They're sometimes off kilter, but we have learned how to regulate drugs and chemicals and other things based on snapshots. 14-day study, 90-day study, 2-year cancer bioassay, and epidemiology again. Trying to get a cumulative response over some period of time and correlating that with exposures, duration, dose, and other things. So if we are turning this back and saying that the microbiome is too complicated and a lot is happening, what can we recommend without going into our session 4, which is the research forward, but what lessons have we learned thus far about, being able to make sense with data we have, rather than just to say that, if we would have had a more granular movie type rather than Polaroid photographs, we could have done better. So is that really a fair question to ask? Is the microbiome that different from other types of studies that we're doing? And what can we recommend to practitioners in terms of them looking for and asking for data like this? I see a lot of puzzled looks. So, but some thoughts are appreciated, because I think a lot of our listeners, again, are practitioners. So we want to help them understand where we are and maybe where we're going.
Okay, well, if I can be provocative, I mean, the first human genome took quite a long time to get and cost a huge amount of money. Now we're talking about the $1,000 genome, which probably means we could get a $5 genome eventually. Now, you only need to get a person's genome once, but what we're talking about in terms of NMR spectroscopy of urine or a bit of LCMS is not expensive. Certainly if you convert it into an industrial process rather than a research one. So if the will was there, I believe we could do metabolic fingerprinting, metabotyping on populations. And there's the beginning of that already. And, so we could take snapshots of people. In terms of precision medicine, one of the visions of my colleague, Professor Nicholson, is the idea of the patient journey, so that when you go into hospital, the first thing that happens is a sample is taken from you. And as you go through treatment, whether that's first on drugs and then surgery and then through the recovery procedure, samples are taken all the time, with the idea of plotting the patient journey through metabolic hyperspace and stuff like that. The idea is hopefully that you will be able to see how a patient is responding and whether they're on a good response pathway or a bad one. And if they're on a bad one, say, whoa, we need to change something well before it's too late. Now, that's metabonomics or metabolomics in a clinical situation. Jeremy is already putting that in place, right? So that's certainly possible and feasible. And Jeremy's also put together what he's calling an Institute of Gut Health, which is looking at the gut microbiome. I mean, the idea of reversing diabetes by turning the gut round and things like that shows you can have some pretty major effects. So I'm quite hopeful if the money is there, an awful lot can be done. Just give me the money.
So Dr. Gerhold will take a question.
Yeah. David Gerhold from NIH NCATS. I was very interested in how the human body and how the gut, for example, encourages the beneficial microbes and discourages the pathogenic ones. And I think Dr. Patterson has begun to address that by suggesting that the bile acids may be regulating it. I think we know that the innate immune system at least also does some of this. But I'm trying to get a grasp on the different ways that the body can do this, and how does it actually distinguish between beneficial and harmful bacteria. And then when we take drugs, for example, immune suppressant drugs, might we be interfering with the regulation that our body is performing?
Okay, I don't want to dominate the discussion, but one of the things I would say to you is that the microbiome is an ecology. So it's not one bug living in isolation to another. It's all the bugs working cooperatively to produce the ecology that keeps them going, like a rainforest. So they will, having established with the climax ecology that they have, they will fight off invasive species. That's why when you swallow probiotics, you have to keep swallowing them every day, because they might be doing something beneficial for you, but they're not helping the other gut bugs, so they will get rid of them. That, by the way, is not a fact. That's an opinion. A lot of the things that I say are not facts; they are opinions. So, but remember Dirty Harry.
But does that imply that the body, the human body, is not having a large effect in regulating that in real time?
It must be having an effect on regulating it, but the bugs must be also. I mean, why don't you get huge amounts of bacterial overgrowth? That has to be this collaborative process between the bacteria that are in your gut, that are working with you for their benefit, and your own your own body as well. It's only when you massively disturb it with things like antibiotics that you get the opportunity for C. diff to start overgrowing, and that's when you begin to get problems, because you kill the things you want to keep, and you keep the things that you wanted to kill.
Anyone would like to weigh in on this? No?
Sometimes I think we look at these things, I think, a little bit oversimplistic, because when you're talking about human biological fitness in any of these etiologies, every speaker had these great slides, and you saw all the diseases and the potential connections to the microbiome. But at the same time, we're not there yet, in making these true connections, because again, there's very specific diseases, and the role of the gut microbiota has an involvement. But again, you have to consider the whole human biological fitness in that context. So I think when we're really trying, and this is what we're faced with now, is if we're going to get into any therapeutic value in manipulating the microbiota for that is we're going to have to, carefully develop studies to really answer these questions probably now that we have a lot of this general information. I mean, I can just talk about, we've talked a lot about drugs this morning, but the more I look at this and I take a critical look, just finished the critical look with the World Health Organization on acute versus chronic, it's very different. When you're talking about acute exposure and chronic, so when you're talking about drug exposure, because when it gets into the gut, first of all, when it's chronic, you're getting it every day over a lifetime, you get that exposure, which versus an acute exposure is one dose, so you have dilution, you have binding differences. Also, you have the architecture of as you look at the gut, you have the architecture of the gut, these ecological niches, you have different microbiota in these niches. And so then you have this whole issue of clearance, drug clearance. So there's a lot of unknown questions. The more you're looking, when you start reviewing, documents and trying to get at where we are in the state of the science, you find out that boy, we work in, as you mentioned in your speech, 10 lifetimes. But there are some very open questions that probably can be answered with animal models, like we talked about. Like you could test this, but normally in typical toxicology, we used to throw away the intestine. Now we're fighting over it, in our own institute, So, but you could take that and then you could do those kinds of studies and you could just look, this isn't rat models. You could look to see, you can answer some of these, but it hasn't been, to my knowledge, it hasn't been done yet.
We'll take the next question.
Thank you. This is fascinating and very...
Please introduce yourself.
Oh, yes, I'm sorry. I'm John Arias from the NIH. This is a fascinating and very complicated system, and also from the standpoint of environmental toxicology in humans, it also raises, of course, ethical constraints on the kinds of experiments that can be done. So considering that, obviously an animal model system has tremendous advantage, yet in my mind I question the idea of using microbial communities that are established probably through coevolutionary relationships in animal systems. So I asked the committee, is it possible, since we've heard about the portability of microbial communities between individuals within a species, to humanize essentially animal model systems with these microbial communities that are obviously of considerable interest from the standpoint of environmental toxicology.
Thank you. So humanizing the gut rather than humanizing the metabolism systems. So Andrew, I'm sure you've thought of this with Frank, because Frank Gonzalez has made great strides in making humanized metabolism mice. So have you started thinking about the humanized gut animals as well?
Yeah, so in that case, Yvonne's referring to replacing mouse genes with human genes. And, this has been a very fruitful exercise because it does allow you to determine those species differences between what one receptor may have or its affinity for a particular ligand. There's lots of examples of this. We've thought about doing humanized microbiome or along with humanized mice. This is well beyond my level of comprehension and how to think about it. The simplest I can say that we are doing is replacing it with gnotobiotic mice with single bacteria, just one, monocultures, because it does allow us to assess the potential or the ability of these bacteria to metabolize particular pools of metabolites. In this case, we focus strictly only on bile acids, and even just doing that is quite complicated. It's very surprising, and on top of it all, it's extremely artifactual, quite honestly, because we are talking about a situation where there is no competition. So these bugs will go pretty much wherever, and some will not take up residence, period. So this is something that we're working on, although struggling to interpret, I should say.
And there is no such thing as one human microbiome as well. I think we need to be reminded of that. So I think the combinatorial designs and all the options are a little beyond the current capacity, I think, in the environmental health and toxicology community. And Dr. Redinbo?
Yeah, so we were quite bullish on using the humanized genetic mice, particularly along the PXR pathway, and then downstream of that, and then humanizing the microbiome or pseudo-humanizing it. But but then we really ran into the roadblock of, so you have a mouse that has a certain set of genes that's human, but the immune system that surrounds the GI is still entirely the murine immune system. And you know, as we, and the role of neurobiology, and so, and I really appreciate what you're doing. And in fact, I'm going to suggest maybe that we work together, but I worry about drawing too much from that, because you know, you're certainly going to have to get to the human, or at least into a larger mammal as soon as you can. And so these are things that we're doing. We're going to the hospital and we're going to enroll patients that are going to take a Reno-Tcan. And then I'm going to make a bold prediction that I can tell ahead of time who's going to have the worst toxicity by what's in their GI. And so, you know, and that's, again, very reductionist and very binary, and so Ian will like that. But it's, you know, it's the sort of thing where we can just get out into the dirty system. And I'll make one more comment about why I can't sleep, and then sometimes how I get to sleep. So I worry all the time about how complicated this is. But then I roll over and fall asleep when I realize that we've approved drugs for decades, you know, without knowing about this stuff. And look, really, and safe drugs, effective drugs, no drug is perfect, but you know, the more we know, of course, the better things are gonna go. But we're, you know, and I could sit here and say, you know, I'll be a Democrat. We should have unlimited funding and we should learn everything. And then I'll be a Republican. I deny that the microbiome exists. And so somewhere in between, we're going to have to find a way forward where we can't do everything. We're going to have to push certain things, and we'll keep illuminating as we go, and hopefully continue to approve safe compounds.
I think I appreciate the binarization of this, and I would probably use a slightly different comparison. I think when you cannot sleep and worry, you're more like an epidemiologist. When you realize we've been, you know, doing okay, you're like a toxicologist. So I think toxicologists sleep a little better than epidemiologists. But Kun, in your studies, you know, have you started thinking about, you know, not just going beyond, you know, recording what effects different exposures have on the microbiome, but more of the, you know, modulating the microbiome with probiotics or some other types of, you know, tertiary exposure, so to speak, where you have your chemical, you have your natural microbiome, and you're trying to maybe add additional factors?
Yes, we have an ongoing study using the food component to modulate the gut microbiome. It's actually the effect actually is very dramatic. You can completely and easily modulate some well-known beneficial species. And right now we are checking whether those microbiome modulation can lead to the change, the arsenic and other xenobiotic metabolism change. So hopefully we are getting there.
Well, we'll take one more question from the floor.
Okay. Ann Summers, Georgia. I want to offer a positive note and also touch on several of the themes that have been mentioned, including longitudinal studies. By describing the classical study, I blush only slightly to say that we did in 1993 to show that common exposure in an animal model, primate model, and over the time of this work we did about 8 different animals and had them in-house in the vivarium over periods of from 2 months to 6 months. And they were exposed at millimolar levels in the GI tract to a very widely found and very widely used therapeutic intervention called dental silver fillings. And it resulted, and so this comes to the point of it depends on what you're looking at. And we saw very profound changes because we were looking for mercury-resistant bacteria, which is a large number of proteobacteria, and also the antibiotic resistance changes. And they were clear that when the fillings go in, you get an increase in mercury resistance and antibiotic resistance and so forth. So the easy part was we were looking at something that is, at the time, we already knew that that's what we could expect. But it was very valuable to see what was really going on by having these longitudinal experiments in primates. And nowadays, primates are actually harder to work with than humans, so I don't think that they could necessarily be replicated, but the value of that. So later on, when we finally accumulated enough money to do this, we looked at the changes in the bulk flora because we were fishing out our Enterobacteriaceae for this experiment. And we looked at the bulk flora in an 06 paper. And there it was very clear in the 4 animals that we had available for that. And this was with classic, I mean, very old, by now standards, 16S RNA kind of profiling. And they were all different. All these animals, 2 were in the vivarium at the same time, and a couple were unrelated. But this kind of thing, just like Kun and others have shown, is just idiosyncratic. And so in this, but we were able to find some correlations between some bacterial populations in terms of a response to mercury. But you can change some things in ways that you can really notice, but other things are going to be harder to see. Now, the caveat I want to add to this is that approximately 30% of the population of humans in the US that are going to be in any clinical trials are going to be similarly exposed to this particular toxicant. And the vexatious problem of any kind of toxicology certainly needs to be kept in mind with respect to co-exposure, and that happens in the gut too. So, thanks.
Well, I appreciate the comment. We had a couple of questions on the web related to the third bullet that we've been discussing already on the animal studies, and I don't think we've had any mention of this. So it's very well known that the animal diets vary in composition of different things, and one of the easiest things that people have recognized is the percent fat, trying to use 4% versus Western diet 30% fat. But there's also other things in the diet. There are purified diets, there are grain-based diets. To our panelists' knowledge, has there been any work to look at the diet composition in the same facility, the same strain, whether that affects the microbiome and in which way? It probably does. Everything affects the microbiome. I think I get that message clear, loud and clear. But what type of an effect that has, if we're not looking at fat, but just what's in the diet per se?
Yeah, I know for sure that groups like Andrew Gerwitz's group in Georgia is looking at this. Georgia State, also at Penn State, his former postdoc Vijay Kumar is also really interested and has published on how diet composition can dramatically influence microbiome. The recent papers about how emulsifiers that are common in our diet, our processed foods now, can impact microbiome with pretty profound effects. So, I mean, those are the ones I can think of off the top of my head.
So, Ian, and in your experience with, CTL and AstraZeneca, how carefully the preclinical studies were about, standardizing, that particular aspect of, of a particular drug or across drugs, across studies over time?
Okay, well, one of the problems when you buy a batch of diet, it specifies how much fat, how much protein, how much carbohydrate, but it doesn't always specify what the origin of the protein was. Now, once upon a time, long, long ago in the last century, when I was a much younger man, we suddenly noticed that the metabolome of our rats had switched from producing hippurate to producing another aromatic hydroxyphenyl propionic acid. And we thought 2 things immediately with horror. One was that we'd lost the ability in our rats to glycine conjugate, which would have been pretty massive. And secondly, that we didn't know what was going on. Now, it turns out that when you switch the diets around between these animals, you could get back to the hippurate producers. So the microbiome was probably the same, but the diet was modulating the production of the stuff in the microbiome. And I've got some nice slides which I didn't show that show a rat starting off with a hydroxyphenyl propionic acid producing urine. You put it into a metabole and it switches overnight to a hippurate producing thing and then switches back. Now, that's microbiome can't possibly change that quickly, can it? But its phenotype has. So again, I hate to go on about complexity, but but it's complex.
Well, thank you. So just to wrap up, I wanted to give all of our panelists one question to answer, and if you would name one thing that is the most important in designing a study of microbiome and a chemical exposure, what would that be? So we'll start with Dr. Lu.
Sample size.
Okay.
Controlling for different effects between your control and your test groups by designing the experiment sensibly.
Cohabitation, or single cage housing, because you can't just simply round robin chemical animals between cages.
If I had a good solution to this, I would have implemented it.
Matt?
I guess funding aside, I would say 2 different sites. Yeah, two different sites for the animal studies, so that the issues of each site's vivarium, different vivaria at the same site.
Replication, basically,
Yeah.
right?
Yeah, your superhuman power stole my answer. That's exactly what I was going to say.
In our human studies that we're conducting, then obviously it gets into statistical power. It has to be, because you have this, we've heard it today, and all of the talks about this inter-individual variability. So without a doubt, that's a key. And then when it comes to the animal studies, then certainly you have to take into account the good laboratory practice. So if that's being considered, then it's just it gets into, believe it or not, 2 points when you're talking about microbiome. One is the handling of the samples. People don't realize the good biology that you have to take when you take that. This goes back to my training with Ralph Wolf and Hungate. I go back many years in getting very traditional training in anaerobic bacteriology, but you have to have really a good sample, because that's going to affect the microbiota. And the second thing, and this is on the other end, getting into where we are today, is on the high throughput sequencing, how that sample is isolated, and then that analysis, because that also, and how that's interpreted, that makes a big difference in all of those charts that we saw today, because we really don't know how it's, I assume it was all done similarly, but if you're making comparisons, it's difficult to do.
Well, I'd like to thank our panelists, and hopefully you've enjoyed the discussion as well, so let's give them a round of applause. We will start again promptly at 1:30 Eastern Time. We have a webcast presentation, so we don't want our speaker to wait. There's a cafeteria up here on the 3rd floor, and then also you can, walk out and find your own lunch.
Actually, for the speakers and panelists and committee members and government liaisons who pre-ordered your lunch, well, if you pre-ordered or not, there's a space reserved in room 106 that you can eat. If you pre-ordered, you'll have a box lunch waiting there. If not, feel free to grab your lunch and come down to room 106. And then last thing is, if the lines are too long here, there is a McDonald's around the corner.